I think we may want to get started. If people are on, our session chairs are. Our Vishal.
Yes, I'm
Patel,
here.
you're here. Thank you, Vishal. Thank you for being so flexible. I appreciate it. And your co-chair, I guess we have a full UT Southwestern team. Is Yogi,
Yes.
and I'm hoping that Andy is on.
Yeah,
I heard him. Is he on?
I'm here.
Oh,
Oh,
perfect.
great.
Okay,
Should
let's see, okay, so I guess we can, why don't we just go ahead so we stay on time? And
Sounds
thank you, Dr. McMahon. Sorry that I didn't make it like completely clear.
Not at all. It's entirely my mistake. I should have realized that the world doesn't revolve around the West Coast.
Unfortunately.
It revolved around the East Coast, actually. Anyway, I'll get started. My name is Vishal Patel. I'm a professor of medicine at the University of Texas Southwestern Medical Center. It's my honor to chair this session with Dr. Yoshiharu Muto, who you will hear in a bit from. I'm going to start by introducing our first speaker, Dr. Andy McMahon, who needs no introduction, but nevertheless is a Merkin research professor at the California Institute of Technology. Andy's research focuses on identifying mechanisms of mammalian kidney development and applying those insights to engineering of functional human kidney cells and systems. So Andy, we're really excited that you're here, and take it away.
Yes, so first of all, let me reiterate my apologies for messing up the schedule and thank the organizers for the opportunity to share our research today. So I'm going to talk about mapping diversity and development of the mammalian kidney through spatial transcriptomics. And really, this lies in the context of the broad interest in the lab in regenerative medicine applied to the kidney. And so for regenerative medicine, we need to identify the cell types of the kidney and their functional organization, identify the underpinning developmental mechanisms, and understand repair processes and the limits to repair. And that's what's been going on in the lab for the last several years. And now it's really consolidated around efforts to generate de novo functional kidney structures, which we see as being useful for disease modeling and therapeutic screening, disease prevention, for example, avoiding drug toxicity, generating renal assist devices with specific cell types, and then ultimately generating functional kidneys, complex composites of different cells. So our work started, and I'm going to focus at the single cell level, with efforts to deconstruct the mouse kidney through single cell approaches aimed at looking at cell types, cell organization, developmental relationships, and focused on both male and female mice. And as you know, the kidney is built along a radial cortical medullary axis of function, and the non-vascular unit of kidney function is shown here, which comprises the nephron in gray, which forms a patent luminal connection to the collecting system in blue. And so given the prominence of cell types here in mediating kidney function, we really focused on these two parts of the epithelial renal tubule network. And so the approach that we took is recognizing that the kidney has this radial axis of organization, was to microdissect into three different zones: cortical, outer medullary, and inner medullary zone, from male and female mice, and using 10x chromium, perform single cell RNA sequencing, informatics to analyze that and validate the data. And we've gone on and done functional studies based upon this data, but I won't review those today. So just to show you some highlights of this initial analysis pioneered by Andy Rancic in the lab. So first, one very obvious feature of the data was the predominant sexual dimorphism that had originally been pointed out by John Rinn in microarray studies in the early 2000s, specifically within proximal tubule segments. So the proximal tubule comprises 3 different segments. And if we look at male and female single-cell RNA-seq data, they basically overlie in all the other cell types within the renal tubule network, but separate very nicely in the proximal tubule regions. And we've gone on in studies with Ivy Zhong and Jing Liu to really try and understand better the basis of this sexual dimorphism in proximal tubule cells. And you can see that if we look at 0 to 2 weeks of development, there's no sexual dimorphic separation, but it starts at around about 4 weeks. And then by 8 weeks of development, then we can see a good separation of the sexes. And here are the numbers of genes that are distinctly female or male biased at those times. So we can see bias at 4 weeks when puberty is starting, and then we can see that there's a major bias at 8 weeks, and that's maintained, although there is some loss in the male specifically of sexually dimorphic gene expression with age. And then if we look at the basis of this, and this has got a lot of different experiments on here, but broadly, this is the female signature of genes female enriched, and this is the male signature of male enriched genes. If we look at the female, if we remove estrogen receptor from those proximal tubule cells or remove the ovary from the female, none of those things have a marked effect on female expression profiles. The only thing that does alter the female is if we inject testosterone, and that will actually partially masculinize the female. In contrast, if we look at the male signature, if we remove the testis from the male, or if we remove androgen receptor from the male, this is removing the testis, this is moving the androgen receptor from the proximal tubule cells, then we can see that this leads to a feminization. And if we inject, after removing the testis, testosterone, we remasculinize the mouse. So this suggests that androgens, particularly testosterone, acting directly on proximal tubule cells, is the major pathway responsible for masculinization. This just shows what some of the data looks like. So if we look at SLC01A1, you can see that in the S2 segment, it's specifically within the male. But if we remove androgen receptor from male proximal tubule cells, we lose expression. And if we look at ABCC3, which is particularly expressed in the proximal tubule S3 segment of the female, you can see a partial feminization in males on the release of androgen, the removal of androgen receptor. So we get now de novo ABCC3 expression in the S3 segment of the male. So this basically summarizes the data that our 2 non-sex organs, the two main non-sex organs that show sexually dimorphic expression, the kidney and the liver, actually have a different mode of regulation, which I find quite surprising. Testosterone acts directly on the kidney and indirectly on the liver through the pituitary and the regulation of growth hormone, which is the direct mediator of sexual dimorphism in the case of the liver. Androgen receptors in the kidney work as directly on regulating gene activity. And interestingly, you all know that in the mouse, males are significantly more susceptible to proximal tubule injury, and resilience comes from caloric restriction and hypoxia conditioning. And in both of those cases, it leads to a feminization of the kidney's proximal tubule expression profile. And caloric restriction actually leads to a drop in testosterone levels, suggesting maybe a direct link there between testosterone and the susceptibility to proximal tubule injury. Another highlight of this is the spatial diversity in nephron segmentation has this temporal link. So the very first of the nephrons produced have long loops of Henle that go deep into the inner medullary region. And those loops of Henle have 5 to 6 different segments within those that are molecularly distinct. And then the late forming nephrons just have a simple thin limb with just one cell type within that. And then if we look at the distalmost part of the nephron, where that nephron connects to the collecting system, really it has indistinguishable cell types from its adjacent collecting system. So it has intercalated cells, principal cell-like cells within this segment, suggesting that the distal part of the nephron undergoes a sort of lineage convergence to resemble the adjacent collecting system cells. And these derive from 2 completely different lineages that arise at different times and places within the developing embryo. And in conjunction with this, preceding this, is the activation of transcriptional regulators, particularly GATA3, within this distal connecting segment, that is a component of the distalization of that region and underlies a component of the expression profiles that are seen in the generation of these different cell types. We can also see novel cell types from tertiary clustering. So, for example, if we look at the distal convoluted tubule and SLC12A3 positive cells, we can see that a subset of these cells also express creatine kinase, an enzyme associated with energy metabolism. So this is present predominantly in the ascending thick limb, but here subsets of distal convoluted tubule cells also express creatine kinase. So all of this is put together in a database which everybody can access. If you just go to Google and then put in Kidney Cell Explorer, you'll come to this database. And this is an anatomical database, since what we did was take genes that demarcated each of these different clusters, and we then tied together these clusters to make this heat map of gene expression along the entire length of the nephron. And there's great studies done by other groups that have also spatially registered their data. Mark Nepper has been a key leader in this in both rat and mouse studies. So you can put in your favorite gene like prolactin receptor here and see that prolactin receptor is specifically within female proximal tubule segments, which suggested to us that maybe something to do with weaning of mice and prolactin activity may be involved in modifying gene activity in nursing females, and this is something that's under study in the lab at present. So our real interest now is in developing human kidney cell types from stem cell-based model systems. And I want to switch gears now to the developing human kidney. And we can see that there's some very obvious features that are different between the two systems. Distinct branching patterns in the developing collecting system in the human, multiple lobes in the human, a much longer period of development. So the period of active nephrogenesis in the mouse is about 10 to 11 days, and in the human, it's closer to 24 weeks. and then many more nephrons in the human, about 1 million per kidney, and in the mouse, it's around about 14,000 per kidney. So this human work initially started in the context of the NIDDK-funded GoodMap initiative, and much of that data can be found at the Atlas website for GoodMap. So, for example, histological, immunological, and RNA-seq data sets that describe human development, and work published in a series of papers from the lab. But I want to concentrate here on single cell and on spatial analysis. And so this is work of Niels Lindstrom in the lab at that time. Niels now has an independent lab in USC, in the stem cell center there. And what we wanted to do here was to develop a 3D anatomy for the human and mouse kidney and compare them, which we did by taking sections, segmenting out developing nephron structures, and performing image registration. And we did this for human and mouse nephrons. And you can see that over the period of nephrogenesis, they look relatively similar. They have a similar sort of patterning. The human is much larger than the mouse, and the human takes about 8 days to go through this particular developmental profile, where the mesenchymal progenitor cells aggregate and then form an epithelial structure and undergo patterning and morphogenesis to this S-shaped body stage. So to understand gene expression and patterning and organization of this key stage, what Niels and collaborators did was to perform immunostaining with 20 regionally diverse proteins to S-shaped bodies, and then to generate a map of protein distribution in the S-shaped bodies. And this is the profile of these 20 different proteins that represent transcriptional regulators, signals, and their receptors predominantly. And then using that protein atlas and inferring RNA expression within cell types from single-cell RNA-seq data from a nephron progenitor cortical enriched precursor population, he was able to then infer where these single-cell data sets lie within this S-shaped body. And now you can see here that we can see particular genes that show positional expression within the S-shaped bodies. And then we can expand from a few genes to show, for example, here in population 6, which is the nascent proximal tubule population, a whole set of genes that are now enriched within progenitor cells for that proximal tubule structure. And if we compare each of these progenitor regions within the S-shaped body, we can see a nascent homology present here with the different cell types present within the adult nephron. So within the adult nephron, there's 22 different cell types within this particular atlas. And we can see that if we look at proximal most cell types within this S-shaped body, 8, 9, and 12, we can see that they have the best homology at this time in their gene expression with proximal cell regions within the adult nephron. But we can still see weaker homology at this time with distal structures in this region. So we can see a nascent organization here that reflects the organization of the proximal distal axis of the nephron, and proximal development is relatively more accelerated than distal development within the S-shaped body. But these are still very nascent progenitor structures. And to sort of go up, and again here, there's an atlas where you can put in your favorite gene, see how its expression lies within the lineage tree, going to those different precursors in the S-shaped body. And then you can see within those different cell types, predicted networks of interacting factors with your gene of interest. So WT1 network here shown within nascent podocytes within that structure. So this gave us a spatial analysis looking at the distribution of genes within the developing precursors of the nephron. And most recently, we've been expanding this to look at the more complete development of the nephron through to functional tubule structures and of the collecting system in data in which we integrated mouse and human single-cell data to be able to directly compare mouse and human development. And as you might expect, human and mouse show similar development as indicated by expression profiles of these genes within united cell types within the developing human and mouse kidney. But we find a large number of genes that are distinctly human. So 1,000 genes show human-enriched cell type specific expression in different populations of the nephron and collecting system. And some of these may actually be of functional disease-associated significance. So, for example, this gene PLA2R1 is human podocyte specific and associated with membranous glomerulonephritis. So here, if you look at the human expression, you can see that it's in this cluster in the human and not in the mouse. And that cluster actually is podocytes. And podocytes are highlighted by MAFB in human and mouse. So red is MAFB and it's present in human and mouse. But PLA2R1 is only present within podocytes in the human, where one might expect it to be, given membranous glomerulonephritis as the associated disease. And we also integrated single nuclear ATAC-seq data within this data sets, and that enabled us to compare putative regulatory regions. And this shows putative regulatory regions that would likely be driving PLA2R1's expression in the human that are absent within the mouse. And these overlap with ENCODE predicted enhancer regions. And indeed, this one overlaps with a single nucleotide polymorphism that is has a strong disease linkage with membranous glomerulonephritis. So the data I've talked to you about up to now is spatial transcriptomics, but in an indirect way, where we are mapping back genes to be able to give us spatial information. The most direct way is to directly take a histological section and map gene expression on that section. And so here we've
5 minutes remaining.
Here we've been collaborating with Long Kai's group at Caltech using SeqFish. And this is an approach that is sequential fluorescence in situ hybridization. And I'll just give you an example of what's being done here in an injury model where we compared injury of the mouse kidney 1 month after ischemia reperfusion injury by SeqFish. And through this approach, we can using 1,000 genes, we were able to map all the predominant cell types within the kidney. We can see features you'd expect. So, for example, the 3 proximal tubule segments distributed as you would expect within cortical regions of the kidney. And we can see vascular cells that are predominantly cortical and medullary that reflect different components of the vascular system. And then we can see that on injury, we get a loss of S3 segments, we get vascular rarefication, we get accumulation of injured cells, fibroblasts, and macrophages relative to controls. And we can do neighborhood analysis and find neighborhoods that associate with perpetual disease state. So this neighborhood 5, which is enriched within cortical regions, comprises populations of injured epithelial cells, fibroblasts, and macrophages. And these are so-called failed repair proximal tubule cells that are highlighted by VCAM expression in the injured epithelial cells, which express the cytokine CLCF1 and CRLF1 within fibroblasts beneath. And these two components are necessary for cytokine signaling through CNTF receptor, which is present within those fibroblasts and macrophages within this pathological niche. So these analyses were able to predict novel injury-associated molecular interactions that might underlie signaling associated with injury. And again, there's a database here that allows viewing of this single cell data. And so now we're moving this seq-fish data to the human developing kidney. I just want to show you a map here that just maps the 4 lineage groups: vascular collecting system, nephron, and interstitial stromal cell types. These are comprised of multiple different clusters, and this just shows them as 4 different lineages within the human kidney. And now what we're trying to do is integrate multi-omic single nuclear ATAC and single RNA-seq data sets back to this registered data set produced with a limited set of probes to get a genome-wide view of spatial expression within the developing human kidney. And so just end there and saying that there's an emerging blueprint for programming of kidney cells, particularly human kidney cells now, and we want to apply this now towards generating functional human kidney cell types. And just to acknowledge my various collaborators in all of this, and those people who are in bold here, all the people who pioneered individual studies that I mentioned today. Thank you very much.
Thank you, Andy. There are no questions in the chat right now. If anybody has a question, please unmute yourself and ask it, I guess. Maybe I can start with a question. Andy, you made a point of showing that the feminization of transcripts in the males happens in the kidney, due to local action of testosterone, but not in the liver, and that happens to the pituitary, the central action. Why do you think those differences exist? Is there a difference in how the androgen receptors are expressed between the two organs, or is there something more complex than that?
Well, it has to be some sort of fundamental difference in the evolution of this. And so it suggests that there's been independent evolution, I think, for this regulatory controls. And I'm not quite sure why it exists, but I will say that it's a more complicated story than I presented today, because we have been looking in some depth now at what goes on in the mouse kidney. And what I didn't say is, as well as prolactin receptor, growth hormone receptor is also specifically expressed within proximal tubule cells. And we've been genetically analyzing prolactin and growth hormone, which are all part of the same family of signals. And there is clearly a role, a direct role, for those signaling pathways also. So the kidney is probably getting multiple inputs, where androgen receptor input is the major control of sexual dimorphism, but it's very likely that prolactin and growth hormone are also acting as well.
Great, thank you. There are a couple questions now popping up in the chat. I'll read them out for you, Andy. First is from Dr. Nuria Pastor Salor, and the question is there any role or findings for GLP-1 receptor in the kidney injury model?
I can't say that I know of anything specific there.
Great, thank you. And the next question is from Brad Yoder, and he's asking, any thoughts on mechanism of how caloric reduction can reduce levels of testosterone and lead to protection?
Yeah, so the connection between caloric restriction and testosterone levels, I don't know, and I imagine that there's people who would be quite interested in that in the longevity business, where this has become quite a interesting area. So I don't know about that myself.
Okay. Gabe, you're here. You just want to ask a question?
Sure. Beautiful talk, Andy. I was wondering, in terms of the mouse-human differences that you're seeing, especially in development, do you know, or do you have a sense from your combined single-cell attack and single-cell RNA-seq, are those driven by differential activity or expression of transcription factors, or is it, you think, downstream of the transcription factors in the way the enhancers and promoters are rewired?
Yes, so I think it's mostly likely novel evolution of regulatory sequences. I think that that's probably what we're going to see, rather than dramatic differences in the expression of the transcriptional regulators themselves.
Okay, great. Thank you so much, Andy. No more questions, and I will pass this on to Yoshi to introduce the next speaker.
Sure. It is my honor to be able to introduce Dr. Gabriel Loeb, who is assistant professor at UCSF. He is a physician scientist focusing on genetic kidney disease. His lab uses cutting-edge technologies and techniques to understand the mechanism underlying common and rare forms of kidney disease. So his lab is focusing on genetic variation in CKD and also molecular mechanism of PKD. So please go ahead.
Thank you so much, Yoshi. Can you see my screen okay?
Sure.
Okay. Thank you so much, Yoshi, for the kind introduction, and for the meeting organizers for inviting me to give a talk, which is not specifically on polycystic kidney disease, but hopefully I'll make a number of connections between the work we've done and I think implications for polycystic kidney disease, which I hope will be of interest to the broader audience. So just by way of introduction, my lab is very interested in using a combination of human genetics and experimental models to really dive into the mechanisms underlying kidney disease. All of you who work on polycystic kidney disease have been doing that now for some time with regard to the genes that are mutated in polycystic kidney disease. But the focus of this talk will actually start with this premise. So if you take all individuals, and this is work from Dr. Ali Gharavi's group at Columbia, if you take all individuals who present to the nephrologist clinic with chronic kidney disease, let's say you have 100 of those individuals, about 3 of those individuals will have polycystic kidney disease. Certainly is the most common monogenic cause of kidney disease and kidney failure. About 6 of those individuals will have some other monogenic form of kidney disease. And about 91 out of 100 of those individuals will have a form of disease that is not mediated by a single gene mutation. And yet, in those 91 individuals, there's still a large genetic component to their development of kidney disease. So if you estimate the fraction of their risk that comes from their genomes, it's somewhere between 30 and 50% of their risk of disease is being driven by genetic loci. It's just that it's many loci across the genome instead of a single locus. And so our lab has been very interested in the question of, can we use those genetic loci to further understand mechanisms of chronic kidney disease? And as I said, I'll make some connections throughout the talk to polycystic kidney disease. There may be those of you in the audience who are somewhat focused or quite focused on polycystic kidney disease and would say, well, he's going to make some connections to polycystic kidney disease, but in general, this talk doesn't apply to me. And I want to convince you that even if you're focused on those individuals with polycystic kidney disease, the loci I'm talking about are actually quite relevant to you. And to illustrate that point, I want to point out some other really beautiful and surprising work done by Christoph Kiriluk's lab, also at Columbia University. And I'll just briefly introduce this by saying what Christoph's lab has done is they've taken all the loci that I'm going to go on to tell you about during this talk, and they've made what's called a polygenic risk score, where they take all of the loci across the genome that are associated with this polygenic risk of kidney disease, and they've basically stratified individuals into tertiles of risk based on their risk loci across the genome. Now, what's known, and Christoph and many other groups have published before him, is if you take random individuals from the population, you stratify them into the individuals with the lowest polygenic risk or the highest polygenic risk. This is showing a log odds ratio of developing or having chronic kidney disease on the x-axis. And you can see that those people at the highest polygenic risk have about a 3-fold increase in the risk of developing chronic kidney disease. So the top third of the population has about a 3-fold increased risk. I think the really surprising observation that Christoph's lab made was that this also holds true for those individuals with pathogenic variants for PKD1 or PKD2. So this is looking at individuals with pathogenic variants for PKD1 or PKD2, mostly actually truncating variants, although they did have some missense variants in here as well. And then they applied that same polygenic risk score to these individuals and looked at the risk of developing chronic kidney disease. And you can see, again, stratified by tertiles, that those individuals at the lowest polygenic risk of developing chronic kidney disease, although they do have a significantly increased risk based compared to the general population, a 3-fold increased risk, had dramatically decreased risk of developing chronic kidney disease compared to those individuals at the middle or highest tertile of polygenic risk, suggesting that all the loci that I'm going to be talking to you about today are relevant not just for the general population who do not have monogenic kidney disease, but also relevant to those individuals with polycystic kidney disease. As an aside, he also showed the same was true for the second most common monogenic cause of kidney disease and kidney failure, which is Alport syndrome. So really interesting work from Christoph's lab, which I think illustrates that the loci that I'm going to be telling you about today are relevant for both the general population of people with chronic kidney disease as well as for individuals with polycystic kidney disease. Okay, so I'm going to divide up this talk. It could be a relatively technical talk, and I'm going to try to make it as non-technical as possible and applicable to the broadest audience as possible. And so tell you about 4 questions we asked, which are, what are the loci across the genome that are associated with the development of kidney disease? What are the particular cell types that are involved? What are the individual variants that are involved? And what are the specific genes that are involved? And I'll say that for a long time, we've been quite good, well, I would say for the last 20 years, we've been increasingly good at identifying loci. But I think we're really, there's really an opportunity for a renaissance in genetic discovery for kidney diseases and other complex traits because we're increasingly, with technological innovation, be able to answer questions about involved cell types, specific variants, and individual genes, which historically have been quite challenging for this field. And so to introduce you to the sort of problem, I want to just plot here on the y-axis a schematic of kidney function, on the x-axis age. And so as nephrologists, we're quite used to thinking about the fact that in our individuals who develop kidney disease, they start out with some kidney function. Over time, this declines either rapidly or gradually. So this would be true, for instance, for someone with polycystic kidney disease. And at some arbitrary threshold, we say that they have disease. And all the genetics I'm going to be talking about today could be done on the binary trait of controls without disease and those individuals with disease. But it's much more powerful to do the genetics on the quantitative trait of kidney function. And so the genetics I'll talk about today is actually done on this quantitative trait of kidney function, but we've validated that all of these loci are relevant for the binary trait of kidney disease or kidney failure, although for the sake of time, I won't show you that work today. You might ask, how can we estimate kidney function? And the way that we estimated kidney function in our studies is the same way I do when I'm seeing patients in the clinic, which is the fact that one of the major functions of the kidney and the function that we're measuring is the ability of the kidney to filter blood. So this is a schematic of the glomerulus that Dr. McMahon was just talking about. And here is a capillary within the glomerulus. And one of the functions of the glomerulus is to filter the blood and small molecules, including the small molecule creatinine or the small protein cystatin C, get filtered out of the blood and go into the filtrate, which ultimately becomes the urine. And in people with kidney failure or kidney disease, the ability to filter the blood is reduced. And so those small molecules or small proteins build up in the blood. And so there are well-validated estimating equations to estimate kidney function as a function of both age and the concentration of creatinine and cystatin C in the blood. And so we estimated kidney function in over 450,000 individuals in the UK Biobank using both creatinine and cystatin C, a combined equation. And with that, we're able to define 430 different loci across the genome that were associated with kidney function in middle-aged individuals. So this GWAS was done in individuals from the age of 40 to 60, humans from the age of 40 to 60. This is a Manhattan plot of those results. What I'm showing you on the y-axis is the association of each individual variant with kidney function measured by both creatinine and cystatin C. And across the x-axis, I'm showing you all the autosomes, so all the non-sex chromosomes of the human genome. For those of you who have looked at Manhattan plots before, you'll notice that this is a quite dense Manhattan plot, meaning many, many skyscrapers here, many, many significant loci. And that's in part because we were able to study this quantitative trait of kidney function. So, okay, so what we had here are 430 loci that were associated with kidney function, and that I told you we validated their role, at least from a genetic perspective in human kidney disease as well. And we think there are many nice qualities of the particular GWAS we did, which for the sake of time, I won't go into, but people have been doing GWASs of kidney function for on the order of 15 years now. And I will humbly submit that the number of molecular insights that have been made from these GWASs have so far been somewhat limited. And so we were very interested in that problem of how do you go from a genome-wide association study to really molecular insights into disease? And to do that, we had to answer those next three questions of what are the cell types that are involved, what are the actual variants that are involved, and what are the genes? So let me introduce you to some strategies that we used to address this problem. So this is a typical locus that we identified as being associated with kidney function. What I'm showing you here is about 400,000 base pairs of chromosome 1. You can see about 10 genes here that are within this interval of chromosome 1. On the y-axis, what I'm plotting is the association of each variant with kidney function. Each of these triangles is an individual variant. And what I want you to appreciate is that there are about 50 variants here spread across over 100,000 base pairs, which are all have very similar associations with the phenotype, in this case, kidney function. So you could have two hypotheses, or two potential hypotheses here. One is that all of these variants actually have some role in kidney function. We think that's actually not the case. The other hypothesis is that actually just one of these variants is actually causal in terms of its role in kidney function. And the other variants are associated with kidney function due to their correlation with the causal variant in the population, a property called linkage. And that's actually what we think is going on here. So the coloring scheme here is the correlation of individual variants with this purple variant here in the population. And so basically what this means is that all of these red variants are highly correlated in the population with this purple variant. So if I know what your genotype is at this purple variant, I can predict with high accuracy what your genotype is at these red variants. And so from the genetics alone, it's very hard to figure out which of these variants is causal, unless it's quite hard to actually get to mechanism of disease. So this issue of linkage actually has been an incredible boon for the type of genetic mapping that helped identify the causal genes in polycystic kidney disease like PKD1, PKD2. And one of the reasons for that is that we know a lot more about the coding genome. So let's say you were going to remap PKD1, PKD2 all over again. And let's say you had a family where you're going to do that mapping, and for whatever reason you decided you're gonna spend the money, given that it's increasingly cheap, and just do whole genome sequencing on that family, both affected and unaffected individuals in that family. The first thing that you might do with that sequencing data would be to ignore all the variants that were in non-coding regions. You would ignore all the variants that you identified in affected individuals that were synonymous, that didn't actually affect protein coding sequence. And you would start focusing on those variants that change protein coding sequence. For instance, the frameshift mutations that cause loss of functional alleles in PKD1 or PKD2. And that would, in pretty short order, now let you identify PKD1 and PKD2 in individuals with ADPKD phenotypes. The challenge with this for GWAS is that most of the common variants that are involved in risk of complex disease, like chronic kidney disease, that are non-monogenic, are actually non-coding. And so the problem of identifying the actual causal variants for these complex phenotypes or polygenic phenotypes is to identify which are the non-coding regions that are relevant for kidney disease. So what is the equivalent of the exon in the non-coding genome? And so work from a number of other groups that has been really foundational for our work has indicated has given us a good starting point. And I'll just illustrate that starting point by saying if you take any given cell type in the human body, about 98 to 99% of its genome is tightly wrapped up around these protein complexes called nucleosomes that are built out of histones, and about 1 to 2% of the genome is unwrapped from nucleosomes and participating in gene regulation. So these are elements like enhancers and promoters and other non-coding regulatory elements. And one of the things that makes a proximal tubule cell different than a neuron is the fact that its regulatory elements are completely different. So these non-coding regions that are unwrapped and participating in gene expression are completely different. Same is true for making a proximal tubule cell different from an intercalating cell in the collecting duct, for instance. And so we can map these non-coding regulatory regions using a technology called attack sequencing, which Dr. Muto referred to and Dr. McMahon referred to in their talks. And this is just an example of attack sequencing in proximal tubule cells, which express this lineage-defining transcription factor HNF4 alpha, showing that basically these peaks of accessibility, each of which represents one of these areas of the genome that's participating in gene regulation. So this is the promoter, and these are potential enhancers around HNF4 alpha within proximal tubule cells that are incredibly specific for the proximal tubule. So you don't even see them, for instance, in other tubule epithelial cell types within the kidney. So one thing that we did and has since been or was being done simultaneously by many other groups was to generate a single cell attack sequencing atlas within the human kidney. And this is the UMAP projection, the low dimensional projection of that data. And the main point that I want to indicate here is that we identified all the cell types that you would expect to identify. So we both identified the epithelial cell types of the kidney, so a cluster corresponding to podocytes, to proximal tubule epithelial cells, to the parietal epithelial cells, to the loop of Henle, to the distal tubule and the intercalating cells of the collecting duct, as well as all the non-epithelial cell types you would expect to see within the kidney. So mesangial cells and fibroblasts, myeloid cells and B cells, 3 different populations of endothelial cells, as well as T cells. And so we were able to use that data actually to go back to our GWAS data and ask the question of what cell types were actually participating in heritability of kidney function in middle-aged people and of kidney disease. So to first sort of illustrate this point, I'll show you a really rare locus within the human genome. So this locus looks nothing like the typical locus that I showed you at the beginning. At this locus, again, I'm showing you the y-axis is the p-value for the association between the variant and the phenotype, in this case, kidney function. You can see at this locus, there's a single variant that's strongly associated with the phenotype and no other variants that are in high linkage or high correlation in the population. This particular variant, and what I'm showing you below are the pseudobulk single-cell attack sequencing data for all the different cell types that we identified in our human kidney single-cell attack seq atlas, organized according to their position along the nephron. So the epithelial cells first with podocytes, parietal epithelial cells, proximal tubule, loop of Henle, distal tubule, and intercalating cells of the collecting duct, and then the non-epithelial cell types within the kidney. And what I wanted to show you for this one example locus, unique locus in the human genome, where it doesn't take any fancy work to figure out what the causal variant is that this causal variant lies within a regulatory element that's incredibly specific for the tubuloepithelium. In fact, it's most accessible within cells of the loop of Henle. And you see absolutely no accessibility in podocytes or in the non-epithelial cell types within the kidney, leading to the hypothesis, but that what this variant is doing to increase the risk of decreased kidney function and kidney disease is to modify the effect of this regulatory element, which we believe to be an enhancer in cells of the tubuloepithelium, most likely the loop of Henle. Okay, that's a single anecdote. We're interested in taking a similar approach across the genome. And so the way we did this was to actually identify cell type specific regulatory elements across the entire genome. So what I'm showing you in this heat map is all 500,000 regulatory elements that we identified within human kidneys. Each column in this heat map is an individual regulatory element. There are 500,000 columns. Each row in this heat map is an individual cell type. And the point I want to make with this heat map is that, especially in contrast to gene expression, regulatory elements are incredibly cell type specific. So these are all the regulatory elements. You can see that about 15% of the and I should also make the point that red here means highly accessible or active in the cell type. Blue means not accessible or inactive in the cell type. About 15% of the regulatory elements are active in all cell types, but most regulatory elements, 85%
5 minutes remaining.
Regulatory elements are active in a single cell type, like these regulatory elements, or active in a group of closely related cell types, like these regulatory elements that are active in the tubuloepithelium. And so I'll just say what we did using a statistical genetics technique called stratified LD score regression is to ask the question for those cell type specific regulatory elements, which ones were actually important for the heritability of kidney function and kidney disease. Surprisingly, at least to me as a nephrologist, we saw no enrichment for those regulatory elements that were specific to immune cells, specific to endothelial cells, specific to fibroblasts or mesangial cells. Instead, what we saw was strong enrichment for those regulatory elements that were specific to the distal tubule, specific to the proximal tubule, or shared by all tubule epithelial cells. And to a lesser degree, we saw some enrichment for those regulatory elements that were specific to the proximal tubule. And so what we believe this data shows is that just like in polycystic kidney disease, where the affected cell type, the directly affected cell type is the tubule epithelium, that in our patients with chronic kidney disease, the main drivers of disease are likely to be the tubule epithelial cells, and to a lesser degree, the podocytes, suggesting that those are actually the cell types whose biology we need to better understand to be able to treat this disease effectively. Okay, so I'm going to skip through a bunch of data here to show you one more tidbit, which is I showed you that most of the regulatory elements that most of the causal variants that we are identifying are non-coding. And a major challenge to identifying the function of those non-coding variants is to actually map those regulatory elements to the genes that they target. And to do this, we've taken a number of different approaches, including a functional genomics approach, where we've actually silenced regulatory elements in primary tubule epithelial cells. We can do this in pooled settings now. We've been able to silence 200 of these regulatory elements associated with kidney function and kidney disease in a single experiment, and then read out the genes that are affected. For the sake of time, I'm not going to go through all the data, but we've been able to use this to identify many genes that are actually, we think, novelly involved in kidney function, kidney disease. For the sake of connecting this to polycystic kidney disease, I want to point out that while for many of these genes, we actually don't know the function yet, and so I think that's an area of great excitement for our lab to identify how these genes are actually playing a role in kidney function and kidney disease. We certainly, a number of genes and pathways jumped out at us when we took this approach and other approaches to map the genes that are important at these polygenic loci, including a number of genes that will be familiar to this audience, so PKD1 and PKHD1 encoding fibrocystin, the gene mutated in ARPKD, as well as a number of other ciliary genes, as well as pathways that I think will be very familiar to people who are thinking about autosomal dominant polycystic kidney disease. So to wrap up, the points that I wanted to make are, first, I think increasingly with technology, we can start to leverage polygenic kidney disease genetics to not just understand the loci that are involved in kidney function, kidney disease, and kidney traits, but actually understand the molecular basis of kidney disease. And as I showed the work from Christoph Kirilik's lab, I think that actually is part of the reason that we see such heterogeneity even within family members who share the same PKD1 variant. I showed you that genetic differences in kidney function of middle-aged humans are largely driven by genetic effects on tubule epithelial cells and to a lesser degree podocytes. Again, interesting, I think that this mimics what we see for monogenic disease, where most of the causal mutations are directly affecting either tubule epithelial cells or podocytes. We talked a little bit, and I'm sorry I had to rush through this, about technological advances that are allowing us to systematically map polygenic loci. And then I think another important point, hopefully for this audience, is that genes that are involved in monogenic kidney disease, including, I talked a little bit about ADPKD and ARPKD, but we also see a number of genes involved in ADTKD that come up in our polygenic loci, are likely also contributing to polygenic forms of chronic kidney disease, suggesting that the biology that you all are working on relevant to polycystic kidney disease and other monogenic kidney diseases actually have a much wider relevance potentially to those individuals with polygenic forms of the disease. So with that, I just want to thank the people in my lab at UCSF, my postdoc mentor and advisor, Jeremy Reiter, in whose lab most of this work was done. Other collaborators, both at UCSF, UC Berkeley, and the Laboratory for Genomics Research, funding sources. And with that, happy to take any questions. Sorry for running a bit over time.
Thank you so much for a beautiful talk and data. So your data is very convincing. To show single cell ataxic data is very powerful to dissect biological and molecular mechanism of GWAS, especially associated with kidney function. I have a quick question. So, what is the percentage of the GWAS loci you identified connected with EGFR are kind of accessible in single cell ataxic data in the kidney? In other words, I'm wondering whether how many GWAS loci are completely closed in kidney single cell ataxic data.
Yeah, Yoshi, that's a fantastic question. I think the best way to illustrate this is this figure, which I skipped through quite quickly. It's actually, if you do it by loci, it's actually, oh, I'm so sorry. If you do it by loci, it's somewhat sensitive to the way you do the analysis. But if we do it genome wide and not with individual loci, it's not particularly sensitive. So this is the way that I like to look at it. So if we look at just those regulatory elements that are accessible in tubular epithelial cells, they explain nearly 60% of the heritability for kidney function. Coding regions of the genome explain another 10%. If we include podocytes, we can explain yet a bit more. So with podocytes, tubular epithelial cell regulatory elements, and coding regions, we can explain about 75%. That leaves about 25% of loci or of heritability that we can't explain through regions that are open in the kidneys that we profiled. We are quite interested in that remaining 25% in the basically cell states that we have not yet sampled that might explain that. And I'm sure many people here can, off the top of their heads, think about many cell types, including particular injury states or the developing kidney, which likely have basically open chromatin at those loci that would explain the remaining 25%. So we're interested in closing this gap, but hopefully that answers your question.
Or in other organ, potentially.
Yep.
And I found several questions. Dr. Andy McMahon's question. So could you comment on how the ataxic profile of the primary epithelial cells in vivo associated with those of specific cell types in vivo?
Yeah, that's a great question, Andy, and something that we constantly struggle with. So I rushed through this, but we're very interested in actually understanding how variants in these regulatory elements function. I showed you one approach where we're actually silencing disease-causing regulatory elements to map them to the genes that they actually regulate. That's obviously dependent upon the fact that you have a cell type in vitro that mimics the biology of the cell type in vivo. All of that work we did in primary tubular epithelial cells because we searched far and wide for a cell line that would basically have accessible enhancers that were relevant and that would recapitulate the biology. It does not exist. I would say the in vitro cell types are an adequate model. So about 60% of the disease-causing regulatory elements that we identify in vivo are accessible in primary tubular epithelial cells cultured in vitro at early stage passages, so like P1 and P2, or P0, P1, and P2. Organoids are somewhat better. About 80% of them are accessible. And I would say that accessibility is sort of the floor because I think there are a lot of regulatory elements that can be accessible and not active. So that gives you some idea of what the fraction of regulatory elements that can be queried with current in vitro models.
Okay, and a quick question from Anna at KUMC. Is there any database where we could access cell type specific regulatory elements controlling gene of the interest, especially kidney primary cells?
Yeah, there is not yet such a database. Our paper in Nature Genetics that came out last year in the supplementary tables basically shows all the regulatory elements that we mapped. And we have recently, as I said, completed a much larger screen of these regulatory elements, and the accompanying that paper, we will certainly make a database to make it more searchable and usable for the community.
Okay, thank you, Gabriel. Wonderful
Great.
talk.
Thank you. Pass it on to Terry. The
Yeah,
music has played.
thank you, everybody. Thank you to the speakers. Great session. We're going to take a 10-minute break. So we're right on schedule. We'll come back at 2:30 and that'll be our last session. We're going to be focusing on clinical science. Thank you, everybody. 10 minutes.